The Somatic Cell Genome Editing, or SCGE, and National Institutes of Health Common Fund logo appears with websites nih.gov and commonfund.nih.gov. A transparent human body is shown with organs inside.
Thousands of human diseases are caused by changes or mutations to the body's DNA.
Examples of genetic diseases are displayed next to the organs affected by the mutations. Huntington's disease in the brain, cystic fibrosis in the lungs, Duchenne muscular dystrophy in muscle, hemophilia in the liver.
What if we could treat all these diseases by diving into our living cells to correct the mistakes?
Animation zooms into the body and moves through a cellular space towards a central cell. Passing into the cell, we move towards the nucleus.
The Somatic Cell Genome Editing program aims to make that happen.
Animation passes into the nucleus. A protein moves towards a highlighted region of DNA that represents a mutation.
Recently, researchers have made great progress in correcting DNA mutations using a technique called genome editing.
The protein cuts the DNA at the mutation, then moves away from the severed DNA. Repair proteins then rejoin the DNA such that the mutation is gone.
And the first tests of genome editing for human diseases are starting. However, there are still some challenges to achieve safe and effective genome editing in patient cells.
Animation transitions back to the transparent body. The organs are highlighted to depict an analysis for genetic changes or mutations.
For example, the DNA in millions of cells needs to be corrected to treat many diseases. And getting the editing tool into different types of cells, such as those of different organs, will require tailored delivery techniques and packages.
An area in each of the muscle, liver, brain, and lungs is highlighted. Examples of genome editing tools, including the protein from earlier, are shown targeting the previously highlighted areas. The editing tools fade off and are replaced by possible delivery packages of various shapes and materials.
The delivery techniques must be specific to enter the right cells and avoid the wrong ones, such as the cells of other organs or reproductive cells like eggs and sperm.
Animation returns to inside the nucleus. A genome editing tool is bound to a region of mutated DNA. The tool cuts the DNA and moves away. DNA repair proteins move toward the break and repair it.
Additionally, different types of editing tools may be needed to correct different types of mutations.
Move to a different editing tool that changes a single base pair mutation on the DNA without cutting it.
The Somatic Cell Genome Editing Program, or SCGE, was launched by the NIH Common Fund.
The DNA environment comes back into focus. An editing tool corrects another DNA mutation.
To develop quality tools to perform safe and effective genome editing in human patients. The SCGE program will make more genome editing tools available to researchers.
The editing tool and corrected DNA are joined by other possible genome editing tools.
Develop better packages to deliver the tools to the right cells.
The genome editing tools and text fade and are replaced by different delivery packages.
Design new tests for the safety and efficacy of genome editing, and make all of the information available to the scientific community to drive future discoveries and cures for patients.
An organ on a chip on a microscope is shown as an example safety assay. The organ on a chip graphic is then joined by the previous delivery packages and editing tool graphics, which rotate around the transparent body. National Institutes of Health Common Fund logo appears along with websites nih.gov and commonfund.nih.gov.